Greetings. So this particular time I would like to take you through cell transport. It's a step-by-step lecture, which I believe is going to help each and every one of you. So the notes will be shared via a link so that each and every person can actually access them. So before we even start looking at cell transport, it's very important to understand that cell transport actually is through a cell membrane. And you and I know that all cells have a cell membrane, which is also referred to as the plasma membrane or the phospholipid bilayer, and so on and so forth. So basically, the functions of the cell membrane are for the control of cell homeostasis. In other words, it controls what enters as well as leaves the cell. Actually, some textbooks say that the cell membrane maintains the integrity of the cell. Okay, it also provides protection as well as support, especially for the internal organelles that are, in other words, for the internal organelles that are found inside the cell. So a little more about the structure of the cell membrane is that it has got two layers of lipids, or in other words, two layers of phospholipids. Hence, we refer to it as a phospholipid bilayer. And this phospholipid is thought of having an head of the phosphate, and then the tail is actually hydrocarbon. So the phosphate head is polar because of the charge, and then the hydrocarbon is actually non-polar. So the phospholipids, since there are two layers, okay, if you look at the diagram on the right, there are two layers. One layer is where it faces inside the cell, and then the other layer is where it faces outside the cell. So since there are two, this is why we said it's a phospholipid bilayer. It is a phospholipid bilayer. Now, inside the membrane are also proteins, and there are two types of proteins. There are those that we refer to as channel proteins, and we also have carrier proteins. We also have the carbohydrates that are also embedded in the structure of the molecule. So like I said, the phosphate head is charged, okay? Then that one is polar, then the phospholipid are actually non-polar. Then you have the proteins that are there, okay? These proteins are either the channel proteins or the carrier proteins, and then you also have the carbohydrates that are on the part of the molecule. So I thought that is basically what we can talk about the cell. So the cell membrane has got some pores within it, all right? Now, these pores are the ones that makes the membrane to be selectively permeable. Selectively permeable in the sense that it is only the materials that are very small in relation to the pores of the cell that can actually pass through the pores. Okay, so things like carbon dioxide, things like water, things like oxygen, I think things like ammonia and all that, those easily diffuse in and out of the cell easily. But for other molecules, it's very difficult for them to diffuse. So it is very selectively permeable. Okay. So now let's come to the types of cell transports that we have. There are basically two broad categories of cell transport. The first one is what is referred to as passive transport, and then the other one is what is referred to as active transport. So whether transport is active or passive depends on whether the cell is spending energy or not in the movement of materials in and out of the cell. Okay, so In passive transport, we have processes like diffusion, facilitated diffusion, as well as osmosis. And the movement of materials in passive transport is that those materials are moving from a region where they are in higher concentration to a region where they are in lower concentration. So as you can see here, this is a downward movement of materials or down a concentration gradient, okay? So if you look at this person who is standing on the hill, you do not need to provide any amount of energy for this person to move, all right? The moment the person is placed on the tip of that mountain, it will start moving. Why? There's already a concentration gradient. So it's like the molecule already have got kinetic energy of their own. So movement is from high to low. On the contrary, active transport is where a cell spends energy. So in other words, the materials are in such a way that you have got more materials in one aspect than the other, all right? So if you have to move materials from a less concentrated region to a more concentrated region, then you need some kind of energy for you to oppose that force or to counteract that force. And like we are saying here, movement in active transport is against a concentration gradient, okay? So you are moving from low to high. And since you are moving from low to high, then you need some amount of energy to be spent. And this energy that is spent is in the form of what? ATP, which is adenosine triphosphate. And you and I know very well that ATP is actually a nucleotide, and energy is only made available if this ATP is actually broken down. The breaking down of ATP is done by the enzyme called ATPase, but also water can actually break that ATP. And when ATP is partially broken down, it is going to give you ATP, okay? Then rather ADP plus a pyrophosphate and 34 kilojoules of energy. Then complete hydrolysis of ATP will give you 3-phosphate, a base, as well as a sugar, okay? So the examples of active transport are the proton pumps, the endocytosis, as well as exocytosis. We'll look at each of these one by one. So like we've said, passive transport, the cell uses no energy, all right? The molecules move randomly. And in other words, the molecules are moving from an area of high concentration to an area of low concentration. And the movement of these molecules will actually continue until what we call a concentration equilibrium has been reached. So when the concentration equilibrium has been reached, then what that simply means is that the molecules are evenly distributed. Okay, the molecules are evenly distributed. So when concentration equilibrium has been reached, we do not say that the molecules are not moving. No, the molecules are moving, except that there is no net movement of molecules, all right? What is going in one direction is being cancelled by what is going where? In the opposite directions. So when the molecules reach equilibrium, the concentration is evenly distributed. The molecules are still moving, except that what is going in one direction is being cancelled in the other direction. So what we say is that there is no net movement of molecules in such a case. However, the molecules are moving. So like I've said, the first one is diffusion, okay? The first one is diffusion. Then the other one is facilitated diffusion. Now, facilitated diffusion is basically diffusion that is helped, okay? And this diffusion is helped with the help of the proteins. And I talked about the two types of proteins. We do have the carrier proteins as well as the channel proteins. We'll talk about that just in detail in a bit. Then the other one is osmosis. So osmosis is diffusion, but it is the diffusion of water molecules. In other words, it is a special kind of diffusion whereby what is actually diffusing is water and not anything else. All right, so now let's talk about diffusion. By definition, we are saying that diffusion is the movement of molecules from a region where they are in higher concentration to a region where they are in lower concentration. Movement of molecules from a region where they are in higher concentration to a region where they are in lower concentration. Okay? And like I said, the molecules will continue moving until a concentration equilibrium has been reached. And under concentration equilibrium, the molecules are evenly spread out. It's not that they are not moving, they are moving, except that the molecules are evenly spread out. Okay? The movement is random because the molecules seem to have some kind of their own kinetic energy. They don't need to be pushed to be moved, no. The movement is with respect to the concentration gradient that has been created. Okay, so like you can see there, you have molecules that are evenly distributed. Okay, so firstly, it starts with a highly concentrated molecule, and then these molecules start moving about until they fill the entire corner or the entire room. For example, if you spread maybe insect killer in your room, okay, you realize that if you spread it on the, or rather in the corner of the room, in no time, the smell of your insect killer will spread evenly to the entire room. Okay, why? Because the corner where you did place your insect killer is highly concentrated. Okay, so what happens? There is more concentration there than the entire room. So from the more concentrated portion of the room, the molecules will start moving until they are evenly spread out in the room. In the same way, for example, you can get a glass of water, and then you introduce a drop of ink at one point in that glass of water. You realize that where the ink has been dropped, it will be highly concentrated. And then over time, that ink will start moving until it changes the color of water from colorless to the actual color of the ink. So the molecules will move from rather high concentration to low concentration. When it comes to facilitated diffusion comes about, like I've said, due to the fact that some of the molecules may not pass the pores that are already in the cell membrane for two reasons or for three reasons. The first reason could be that those molecules are larger than the spaces that are provided, or they may have some kind of a charge that may limit their passage. So when you look at the two scenarios that have been presented here in A and B, so B is the normal scenario for most materials where they are passing through the intraspaces between the phospholipid bilayers. And like I've said, these materials may have the same size, or rather the size is slightly lower than the pores that are here. They are not charged so that they can probably interfere with the charge on the phosphate heads and so on and so forth. So they're able to pass through this. However, in B, or rather in A, the materials that are not able to pass through the normal spaces passes by means of the proteins. And like I've said, there are two types of proteins. The first one is what is referred to as a channel protein, okay? Like the one that is in A. So a channel protein is just providing the channel through which these molecules are passing. Why? Because they cannot pass through the spaces that are provided by the phospholipid bilayer. The other type is what is referred to as carrier protein. So for the carrier proteins, the material that is being transported gets bound by this protein, and that is how it is transported from either outside or inside of the cell. For the carrier protein, the material is bound. For the channel protein, the protein just provides a leeway, or in other words, it provides the passage through which these materials can actually pass. Okay, so you see the materials are going to pass through the normal channels of the cell membrane, that is diffusion. If the materials are going to be helped, that becomes facilitated diffusion. And this facilitated diffusion is either through the channel protein, where the protein is just providing the channel or the passage through which materials are passing, or through the carrier proteins, where the materials that are being transported are bound by the proteins, and then they are transported either inside or outside of the cell. Okay, so all these are the different types of cell transport, which is actually passive. So the movement is from high to low. So even with the carrier proteins, the materials are also moving from high to low. Next, let's go to osmosis. So kindly note that in diffusion, anything can move. Okay? In diffusion, anything can move. The water can move. Okay? In other words, water, which is a solvent, can move. The solute can also move. Okay, but when it comes to diffusion, what moves, or rather, when it comes to osmosis, what moves are actually water molecules, nothing else. Okay, what moves are basically water molecules, nothing else. That's why we said osmosis is the diffusion of water molecules. Now, these water molecules again are not moving anyhow. All right, the water molecules are moving through a selectively permeable membrane. In osmosis, what is moving are the water molecules, but these water molecules are moving through a selectively permeable membrane. So since it's diffusion, it means that the water molecules are moving from a region where they are in high concentration to a region where they are in lower concentration across a selectively permeable membrane. Okay, then next we go to active transport. So active transport uses energy. Why? We are trying to force people who are, or materials that are less, to oppose those that are many. Okay, it's like in a fight. Okay, if two groups are fighting, then one group has got two people, then the other group has, say, 4 or 6 people. The group that has 2 people must be very powerful enough, okay, to defeat the group that has 4. Why? Because in terms of energy, they are less. So they need to apply more energy. It's the same concept here. So the movement is from low concentration to high concentration. Therefore, energy has to be spent. And the types normally that we consider under here are 1, the protein pumps, 2, endocytosis, and then 3, exocytosis. Okay, the protein pumps, the endocytosis, as well as exocytosis. So if you look at that drawing that is there, you will realize that there is more potassium, okay? There's more potassium, or rather sodium, inside the cell than outside the cell, okay? So the normal should have been that the sodium that is more should go inside, okay? However, the cell homeostasis says that you do not need so much sodium inside the cell, okay? Why? Because that may affect the integrity of the cell and so on and so forth. So the little that is there is still pumped out of the cell. So to do this, you expect to spend some amount of energy. Some amount of energy needs to be spent. Okay. The next one is endocytosis. So endocytosis is when bulk materials, okay, are taken into the cell. So how this happens is that the material to be taken in comes near the cell, okay? Then the cell forms what are referred to as invaginations, and then the cell folds around the material and then it is taken inside, okay? This is actually a type of transport that is basically used by some freshwater organisms like the amoebas, okay? It is also used by the white blood cells called the phagocytes. This is the way in which these phagocytes are actually able to swallow bacteria and other foreign materials that are found or that could be found inside the human body. Okay, so this is referred to as endocytosis or cell eating, or if you want, you're actually going to call it as phagocytosis. Like I've said, this is how the white blood cells, for example, eats bacteria. The other one, the opposite is also true, which is referred to as exocytosis. So the materials are being forced out of the cell, okay? So in endocytosis, materials are coming from outside of the cell into the inside of the cell. In exocytosis, the materials are moving from inside the cell to the outside the cell. The mechanism is usually more or less like the same as it happens in endocytosis. And this is the process rather through which hormones and other wastes are excreted, or in other words, are released from the cell. So these are the types of cell transport. Now, when it comes to osmosis, how water moves, whether in and out of the cell, is dependent on the types of solutions. How water moves in and out of the cell is dependent upon the types of the solution. Okay? However, there are times when the water is sometimes too much or too little, and whether water is too much or too little, or sometimes it can be the same in the two different concentrations, it may have an impact on the cell. It may have an impact on the cell. So that brings me to another type, under this topic, where we're going to talk about solutions. Okay, there are three types of solutions. There's what is referred as to hypotonic solution. There's what we also refer to as isotonic solution. And then the last one is hypertonic solution. Okay, hypertonic solution. So in a solution that is hypotonic, okay, in a solution that is hypotonic, what we are saying is that the solution has lower concentration of the solutes and a higher concentration of water. In other words, it is less concentrated. It is less concentrated. Okay, so these terms can be used interchangeably. For example, if the inside of the cell is highly concentrated, we are going to say the inside of the cell is hypertonic. If it is less concentrated, we are going to say the inside of the cell is hypotonic. So in one environment, you can have hypotonic. In the other environment, you can have hypertonic. Okay? Hypertonic. So if the inside of the cell is less concentrated, then the outside is more concentrated, water tends to leave the cell. But in this case, if, for example, an animal cell is placed in a solution that is hypotonic, so this solution, which is hypotonic, has got less solute compared to the cell. What happens? Water will start moving into the cell. Mind you, osmosis tends to dissolve what is highly concentrated. So the cell is highly concentrated. So water will start moving inside the cell. And as a result of that, the cell swells up and bursts. Why? This is an animal cell. It swells up and bursts. Why? Because there is no strong cell wall to create tagger pressure and then stop the cell from expanding. This was going to be different if this was actually happening in a plant cell. So an animal cell placed in hypotonic solution probably swells up and bursts. Okay. However, when a plant cell, okay, is placed in a solution that is hypertonic, all right? A solution that is hypertonic, meaning the outside of the cell is more concentrated than the inside, okay? So what happens? Water moves out of the cell, causing the cell to shrink, and therefore the cell will die by the process that is referred to as plasmolysis. So plasmolysis happens to the plant cell. Lysis happens to the animal cell. A solution that is isotonic, these are in equal concentrations. So what is inside the cell is same as what is outside the cell. What is inside the cell is same as what is outside the cell. So here there is equilibrium. There is no net movement of water. In other words, water is moving in both directions at the same rate. So this will have no impact on both the plant as well as the animal cell. It will have no impact on both the plant as well as the animal cell. Okay, so this is basically what happens when these cells are placed in different solutions. You can say, for example, for the animal cell, when it is placed in a solution that is hypotonic, it will shrink. Okay, it will shrink because it loses water. The plant cell becomes plasmolized, In an isotonic solution, there is no change because the movement of water in and outside is basically the same. And then in a solution that is hypotonic, the plant cell becomes targeted, okay? Turgidity builds in, and then the animal cell lyses. It gets in a lot of water, and then at the end of the day, bursts or lyses. Okay. Now, what you can now say at the end of the day is that these water movements in and out of the cells have implications on the lives of the different animals. Okay. For example, bacteria and plant cells, they've got the cell walls. Okay, so they cannot expand anymore. So even if you place them in a solution that is hypertonic, okay, they'll reach a place called tiger pressure. So tiger pressure is as a result of the cell wall. Then for protozoa or protista like paramecium, these have got specialized organelles called contractile vacuoles. So these are able to collect and pump water out of the cell. For the fish that stays in water or salt water, they've got specialized gills, and then animal cells are kept isotonic almost all the time so that they do not die off. Okay, so this is basically the end. This is the basic of this, and then the other recordings will follow later. Thanks a lot and all the best.